Hello, everyone. My name is McKenna Stoker, and today I'm going to show you pretty much my research paper, which was on the evolution of HIV. So starting out, we're going to talk about some HIV statistics that I found while I was researching, and it's kind of why I wanted to research this project, is because I was kind of amazed at how big the statistics were for this disease. So as of 2005, there's 33.4 million people infected with HIV, with most of them being in Southern Africa. And since 1981, 28 million people have died from HIV/AIDS, and there's about 14,000 new infections every day, which is crazy to me. And it explains to me why HIV is so well known, and it's a disease that you can mention it and everybody knows somebody or has heard of somebody that has experienced this disease and gone through it. So starting out, I wanted to find out the origin of HIV, and I came across the story of how in the 1980s, researchers started noticing that a lot of the laboratory primates were infected with a disease called simian immunodeficiency virus, or SIV. And it didn't seem to have any negative effects on the primates. They just had a virus living inside of them, kind of as a, what's the word for it? Sorry. It wasn't a parasite, but a symbiotic organism. Later, however, a virologist from the Tulane National Primate Research Center named Preston Marks won an award, and he decided he wanted to talk about the origin of HIV and realized that there wasn't any really recorded history. Preston knew that HIV was a derivative of SIV, the virus infecting the laboratory primates. And so he decided to take himself and his team into the home country of where some of his infected primates came from, which happened to be Sierra Leone. While Marks and his team was in Sierra Leone, they took 10,000 people who had monkeys and surveyed them. And surprisingly, they only found 2 cases of actual HIV within the population. This really surprised Marks and his team because SIV was present in almost every monkey, chimpanzee, and old world primate in the area that they could find. And yet the same virus, HIV, was super rare in the human population. So why is HIV not as rampant as SIV? So after thorough investigation, Marks and his team found that SIV has been in Africa for at least 1 million years, meaning that the virus had time to become highly divergent and infect every species of monkey and specialize to that species. A theory for why HIV isn't as rampant among humans is because it hasn't been able to infect humans for as long as SIV has been able to. To test this, some researchers named Betty Korber, James Thieler, and Stephen Walensky took a blood sample they found from the Republic of Congo, taken in 1959, and they used this to predict if SIV had jumped to the human population by then. And they actually found that the blood sample was infected with a strain of HIV called ZR59, which happened at least in the early 1930s. So we don't know the exact date that HIV did make this jump to humans, but we know it happened at least in the late 1930s. So going further, we have to figure out how the virus had jumped to the human population. Now, there's many theories about how the initial infection took place, but the most likely is that it was through cutaneous or membrane exposure to infected ape blood or body fluids. So bushmeat hunting is a very common practice in West Central Africa, which is what the picture you're seeing is. And in this practice, hunters go out and creatures, and one of the most common types is monkeys. And so the standing theory is that a hunter injured himself while hunting, and their open wound was exposed to the blood of an infected ape. There was another theory that infected chimpanzees' tissues were used to create the oral polio vaccine, which would in turn infect all the people who took it, but this theory was disproven just recently in 2004. So now we're going to move on and we're going to talk about how HIV actually physically moves around and jumps to the human population. So there's two subtypes of HIV. There's HIV-1 and HIV-2, and each one has multiple subtypes in its own category. So it's estimated that HIV jumped from animals to humans at least 10 different times, and that it's very likely that this will happen again. So HIV-1 consists of groups M, N, O, and P that were caused by human to ape cross-species transmission in West Central Africa, which you can see in this map here. Group M and N are closely related. They're actually from southern Cameroon, and the most accepted ape that it originated from is chimpanzees. Group P and O are also closely related, and they are expected to have originated from West Central Africa. We don't know the exact coordinates as well as we know groups M and N. And then finally, Group P is derived from gorillas, but there's been so few strains of Group P that we haven't really been able to get enough strains to characterize what region and when the transmission initially occurred. So HIV-2 is mainly restricted to a couple countries in West Africa, and these are Guinea-Bissau and Senegal. HIV-2 is the most closely related to the SIV infection, and it originally came from sooty manic bees, which is a primate hunted in West Africa. So groups A and B are spread to humans, but groups C through H have very limited spread and have only ever infected a few individuals, so we don't know a lot about them. But we do believe that they've originated from the Côte d'Ivoire, while group D, Liberia, and groups E and F are from Sierra Leone. So moving on, we're going to talk about how HIV actually attacks the host. In the human body, we have specific cells that are made to identify and destroy virally infected cells, and these are called the CD4 positive T helper cell and the CD8 positive cytotoxic T cells. From this point on, I'm going to refer to them only as helper T cells and cytotoxic T cells. So helper T cells have receptors on them called MHC class II, which identify the epitopes on virally infected cells. When one of these cells identifies the epitope for a virally infected cell, it will signal a macrophage to begin to divide and secrete proteins that activate and promote the complement system. The immune system activation then triggers the cytotoxic T cells to hunt down cells with an MHC class I receptor that displays viral epitopes. Once the cytotoxic T cell finds these cells, which are considered non-self, they bind to the epitope and induce apoptosis, which is when they burst the cell wall and kill the cell. So normally in a normal healthy human, these mechanisms are enough to limit and kill infections and keep the host healthy. However, in HIV, the virus actually colonizes the helper T cells. So once they get inside the helper T cells, the virus uses reverse transcriptase to turn its viral DNA into DNA, which is then inserted into the host genome. The infected cell then begins to create new HIV proteins. And then eventually, once the cell runs out of nutrients and supplies, the cell will burst and thousands of new viral particles will be released into the bloodstream, where they will begin the whole process again. So as the number of the viruses increase, the helper T cells and macrophages begin to die off because they're being lysed so quickly by the virus. However, the cytotoxic T cells and beta lymphocytes kill as many virally infected cells and viral particles as they can find. These efforts help the cell combat the onslaught of viral growth and offer a small buffer period where the body tries to restore itself of helper T cells. So this phase is actually called the initial phase of the infection, which you can see on the screen, and this is when helper T cells and macrophages begin to decline. Second phase, the immune system continues to function fairly normally while the level of viruses grows exponentially and the number of helper T cells and macrophages are steadily picked off by the viruses and more cytotoxic T cells. This happens so quickly, however, that the human body is unable to maintain the helper T cell population, and the immune system begins to plummet. Now, this is where we get into stage 3 of the infection, which is typically called AIDS, and it's the last and final fatal stage. So in a normal, healthy individual, a person typically has 1,000 helper T cells per microliter of blood. However, when patients get to this stage, they have only about 200 helper T cells per microliter of blood. Without the helper T cells and macrophages, the viral load skyrockets, while the host system becomes pretty much non-existent. This makes the host a perfect incubator for the virus, while also leaving them vulnerable to normally non-threatening infections, which is what usually causes death in HIV-positive patients. So now we're going to move on. We're going to talk about the evolutionary success of HIV, because it really is an incredible virus. So HIV has a couple mechanisms that allow it to overwhelm and destroy the immune system. These are rapid evolution, co-infection, and organ differentiation. So the major player in this is the rapid evolution. So compared to mammalian DNA, HIV evolves approximately 1 million times faster than our DNA. This allows short generation times of the HIV virus, which in turn lets the virus adapt to the host environment over the course of multiple generations and become more powerful and more destructive and more specialized towards the host. Another thing that helps contribute to this rapid evolution is that HIV's reverse transcriptase is very error prone. And the reverse transcriptase is what is in charge of converting RNA into DNA. And so when this reverse transcriptase makes a lot of errors, it allows point mutations in the genome to occur within every virus. And with so many point mutations taking place, since they do replicate so quickly, HIV has ample opportunities for the virus to gain an advantageous mutation that will help them adapt, evolve, and thrive within the host. Next point of evolutionary success is co-infection. To further the infection in the host, HIV particles have begun to inhabit the same cell in a process called co-infection. So researchers typically thought that one virus particle would go into each individual host cell. However, some researchers found that as many as 3 to 4 viral particles can inhabit each host cell. This allows each virus particle to introduce a fraction of its genetic code into the DNA genome of the cell. This means that the next generation of viruses will maintain the beneficial adaptations of their predecessors and help ensure the survival of the virus inside the host as well as outside the host while it's being transmitted. Co-infection also helps increase the cytotoxic T cell lymphocyte escape mutagens, or viruses that escape the cytotoxic T cells. And these escape mutants are then free to infect new cells and create new viruses and co-inhabit with new viruses that also survive, making the future generations more potent and more effective at dodging the immune system and infecting the host. So the last part of the evolutionary success is the organ differentiation. And I think this is possibly the biggest contributor to how powerful HIV has become. So organ differentiation is this ability for HIV to adapt to each individual organ it inhabits. So some researchers looked at HIV-infected cadavers, and they found that the HIV-1 viral strains infecting the blood and semen of the cadaver displayed different biology than they displayed different biology and an ability to compartmentalize within their organs, whereas they found a different strain of HIV-1 virus that did not resemble the previous one took up residence in the blood and semen. So the HIV viruses in the blood and lungs have shown to follow different evolutionary paths as well. HIV-1 virus is often considered to be composed of independent evolutionary lineages. So we're going to talk about this. We're going to go into more detail with this to help us understand. So HIV-1 viral proteins infect lymphoid or monocytic lineages. or macrophage lineages. So the lymphocyte T tropic strain that uses the CXCR4 co-receptor begins infection, while the macrophage tropic uses the CCR5 co-receptors. So already we're seeing that they use different techniques to invade the body. However, there's more. The lymphocyte T tropic strain of HIV is found in the lymph nodes, blood, bone marrow, and spleen, and which are considered T tropic organs, while the macrophage tropic strains of HIV are found in the colon, testicles, lungs, and the brain. So some researchers wanted to prove that there was a relationship between compartmentalization and organ differentiation. And so to do this, they began examining organs from within the patients to find which codons were being positively selected, or in other words, seeing which genes were being expressed within these HIV particles. They actually found that the codons that were under the most positive selection and compartmentalization were 48.9% more frequent in their prospective organs than what the researchers expected to find. This ability of the HIV strain to compartmentalize in specific infection sites supports the idea that selective pressures of different organs affects the means by which HIV enters, reproduces, and thrives inside of each different host organ. One of the explanations for organ differentiation in HIV is that each organ has a different susceptibility to antiviral drug penetration, meaning that they're not affected by drugs in the same way and that drugs aren't able to work the same way in them. According to this theory, only the strains of HIV that have the advantageous mutations are able to survive in organs that are more susceptible to drug therapy, while the strains with less mutations that are less advantageous can only survive in organs that are more resistant to the influence of antiviral medication. So I know that was a lot of information, but I'm going to do a quick little recap that'll hopefully help clarify some things for you guys. So as we said before, HIV is probably one of the most well-known diseases, and for good reason. It has incredible statistics when you look at it. For years, we knew very little about HIV. However, since we made the connection between HIV and SIV, the virus from West African primates, big evolutionary changes were discovered. We discovered that HIV has made multiple jumps into human hosts, and that multiple strains were created and these strains can be used to teach researchers and doctors how to control and prevent the spread of HIV. Learning how HIV reproduces, thrives, and conquers the immune system of the host by rapid reproduction, co-infection, and organ differentiation can provide an insight to researchers and doctors as to how to treat the disease and contain the infection. Once we understand all these mechanisms of HIV's evolution, doctors and researchers will finally be able to establish a vaccine, a cure, a medicine plan, or a treatment plan. Thank you for listening, guys. I know it's a lot of information, but hopefully you found it as interesting as I did.